We have Somya Prabhu from Manipal School of Life Sciences in India.
Yes, ma'am. Am I audible?
Yes, go ahead.
Malaria, the most ancient of the deadly infectious diseases, affecting millions of people, is the cause of serious illness and death of young children and pregnant women. The transmission of Plasmodium through mosquitoes causes malaria, with Plasmodium falciparum being the deadliest. Though after showing a declining trend over the years, a sign of reversal has been exhibited for the year 2020, owing to the disruptions caused to the services due to the COVID-19 pandemic. Hence, it is crucial to identify the challenges and propose solutions to maintain the declining trend in order to achieve zero malaria. As the clinical manifestations appear with erythrocyte infection, few resistant phenotypes like sickle cell anemia, garbage negative phenotype, ovalocytosis have been documented. Prevention strategies that include vector management approaches such as use of insecticides, bed nets, and development of effective vaccines. And early diagnosis is essential for reducing complications, and the major classes of drugs that are used to treat malaria include artemisinin derivatives and quinoline-related compounds. Unsurprisingly, over the years, Plasmodium has evolved to resist treatment using antimalarials due to the factors such as improper dosing, incomplete treatments, use of fake drugs. Overall, host immune response, parasite species, their variants, host genetics, and treatment strategies all contribute towards the outcome of the infection. Hence, identification of such contributors to disease progression is an ultimate challenge. MicroRNAs are emerging as very powerful genetic regulators, as evidenced by the fact that a single microRNA can direct entire cellular pathways via interacting with a broad spectrum of target genes. This property renders microRNAs as highly interesting therapeutic as well as diagnostic tools. MicroRNA levels are thus being modified with the use of either mimics or inhibitors, and they have been demonstrated to play a major role in wide range of diseases, though underexplored in malaria. As already known, every stage of hematopoiesis has been shown to be regulated by microRNAs, like MIR451, MIR144, which is essential for erythrocyte proliferation and differentiation, MIR16 for hemoglobin synthesis, and MIR142 for maintaining the erythrocyte morphology. By the very fact that sickle cells resist plasmodial infection due to low oxygen availability, structural changes, as well as differential microRNA activity that was shown to inhibit parasite proliferation. Hence, investigation of such host-based therapeutics is promising. Role of miRNAs in regulating malaria and its varying phenotypes have been documented in literature, though their role to be used as therapeutics is yet remain to be explored. Plasmodium falciparum erythrocyte membrane protein, which is one of the key factors responsible for rosetting and cytoadherence, contributes towards pathogenicity and immune evasion of Plasmodium falciparum. Each variant is unique to the disease type and it possesses distinct adhesive properties. So targeting this virulent gene, which is present in about 60 copies in the genome, is an ultimate challenge. Towards this, we have performed an integrated analysis by utilizing the Plasmodium EMP transcripts and microRNA expression datasets. We did identify a subset of microRNAs which were predicted to strongly target the EMP gene. So though tools for targeting EMP that is highly variable is unlikely, but identifying the significant variants amongst the thousands circulating in the population is an ultimate challenge.
All right. Yeah, thank you. Next, we have Ingram Leslie Ingram from the Institute of Medicine and Medicinal Plant Studies.
Hello, can you hear me?
Yes, go ahead.
Thank you. Greetings from the Institute of Medical Research and Medicinal Plant Study, all the way from Cameroon. Malaria caused by 5 Plasmodium species remains one of the global threats to health and well-being, with the WHO African region accounting for the highest proportion. Malaria in endemic countries like Cameroon is often manifested as a function of immunity that comes with age, reason why asymptomatic malaria is always assessed in aged children and in adults, leaving behind vulnerable groups like children under the ages of 5. Even though asymptomatic malaria might be beneficial in inducing and sustaining a partial immunity against malaria, it might give rise to severe disease complications characterized by coma, severe malaria, especially in vulnerable groups like children under the age of 5. Reason why we conducted a study on the prevalence of asymptomatic malaria carriage in children under the ages of 5 in Yaoundé, Cameroon. In order to do this, we use a species-specific heredity light microscopy and PCR-based survey, where we used the different methods to survey malaria prevalence in 127 children. These species-specific survey methods enable us to identify a significant number of malaria parasite in children and in asymptomatic children. For instance, when the children were assessed from asymptomatic malaria using the rapid diagnostic test, 95% of the 127 children assessed were tested positive for malaria. When that happened with the light microscopy, 71 of them were tested positive for malaria, and with PCR, 85% were tested for malaria. We also try to look at the sensitivity and specificity of light microscopy and rapid diagnostic tests with respect to PCR that we consider as a gold standard in this study. And we discovered that the sensitivity of light microscopy and the rapid diagnostics tests were almost the same, whereas the high positive predictive value and negative predictive value were observed with rapid diagnostic tests as compared to light microscopy. We also did a speciation in order to identify the different predominant species that were causing asymptomatic malaria in these children. And we found out that Plasmodium falciparum was the most dominant, followed by malaria and then ovale. Vivax was absent in a study. Then from all that, we concluded monitoring changes in the number of asymptomatic malaria cases is useful for measuring malaria outcome and putting up intervention, especially in most vulnerable groups, such as children under the ages of 5. Thank you.
Thank you. Next, we have Tosin Okbadokun from, sorry about that, from McGill University.
Thank you, Abir. Can you hear me?
Yes, we can.
Okay, so so Plasmodium falciparum infected red blood cells release extracellular vesicles, which are implicated in parasite biology and disease pathophysiology. Now, these EVs are small particles which are released by viable cells, and they contain proteins, nucleic acids, and lipids, which they deliver to target cells to induce a response. Now, despite the increasing interest in EVs in malaria, the isolation of these particles is not standardized, and EVs that are released by red blood cells infected with the erythrocytic stages of Plasmodium have not yet been fully characterized. So to address this challenge on the one hand, as well as fill the knowledge gap on the other, our lab has developed an optimized and technically reproducible differential centrifugation protocol for EV isolation from red blood cells and P. falciparum infected red blood cells. And we have comprehensively analyzed these EVs. Using nanoparticle tracking analysis, we have observed that the EV subpopulations, which we have designated pellet 10 and pellet 100, are mostly 200 nanometers or below in diameter. And the particle concentration of uninfected red blood cell EVs is much lower than that obtained from the infected red blood cell EVs. And within this infected red blood cell EV populations, the P100 vesicles have a significantly higher concentration than P100. What's interesting, however, is that the Western blot analysis of selected red blood cell proteins show a greater intensity of these proteins, namely the glycophorin A, flotilin 2 and band 3 in P10 compared to P100 EVs, suggesting that despite being less populous, the P10 EVs are loaded with a lot more protein cargo than P100 EVs and are therefore denser. Mass spectrometric data of band 3 corroborates our Western blot data, and mass spectrometry also shows that the EV subtypes, that is P10 and P100 EVs of the ring, trophozoite, and schizont infected red blood cells contain both red blood cell and parasite proteins that are unique to them. And so we conclude that red blood cells infected with the erythrocytic stages of P. falciparum release a significantly higher concentration of EVs than uninfected red blood cells. And these EV subtypes and subpopulations have distinct protein profiles. Our findings are very crucial for the design, the analysis, and application of functional EV studies. Thank you.
Thank you so much. Next, we have Alessandro Grosso from the University of Antwerp.
Hi all, can you hear me?
Yep.
Obtaining reliable estimates of malaria transmission intensity is difficult due to the dependence of the transmission cycle on a wide range of factors interacting with each other. Central to my work is arguing in favor of using the force of infection, which counts the number of new infections per person per unit time, as a metric for malaria transmission. From a statistical perspective, estimates of the force of infection can be obtained directly by modeling parasitemia or serological data, thus avoiding costly and time-consuming collection of entomological data. Methods such as GEEs or GLMMs, dealing with observed and unobserved sources of heterogeneity, are an option, as well as nonlinear models such as GAMS. From a mathematical modeling perspective, the force of infection is expressed as a parameter tracking the movement of the human host out of the successible compartment. Its use leads to a notable simplification in the complexity of the associated system of equations, which would not have to explicitly model the vector, now represented implicitly by the parameter for the force of infection. Last, studies normally proceed by analyzing mathematical and statistical models separately. However, one might not have a full picture of all factors to include in the equations describing transmission. And at the same time, statistical models rely on assumptions about the data generating process, and formulating these assumptions is much easier and transparent when this can be derived from a mathematical model supported by the best biomedical knowledge. As a result, there is much to gain from combining the two approaches, by first expressing a set of compartmental mathematical models, then proceed by either indirect parameterization, expressing a parameter of interest as a function of other estimable quantities, in turn estimate these quantities with a series of statistical models, and then replace the estimates back into the mathematical model, or proceed by direct parameterization, by positing a mathematical model directly for the force of infection and include this model in the likelihood function. Last, one could iterate between the mathematical and the statistical models to identify the best fitting model from a set of theory-derived equations. To conclude, my research provides a general framework for the analysis of malaria data, encompassing robust, precise, yet flexible methods which analysts can employ to support both ex-ante decision-making and ex-post impact evaluation. Thank you very much.
Thank you. I think we missed one. Joseph Sunday, are you on? I still do not hear. Oh. Bojo Sunday, I can see you in the participant list. Unfortunately, we can't hear or see you. Oh, we can hear you, I think.
Oh, yeah, thank you. Thank you for having me. I'm Sunday Uju. I'll be talking on prevalence of mutant dihydrofolate reduces DHFR gene, and it's sorry.
You can go ahead.
Hello.
Yes, we can hear you. Can you hear me?
Hello. Hello.
Hello?
Yeah, can you hear me, please? Hello.
Yes, we can hear you.
Yeah, I'm not sure. I'm not sure you can hear me. Can you hear me? Oh, thank you. Yeah, thank you, everyone. I'll be talking on prevalence of mutant dihydrofolate reduces gene and its implication on permitting resistance among asymptomatic Plasmodium affected adolescents in rural communities in Kwara State. Of course, as we know, malaria causes morbidity and mortality worldwide, particularly in sub-Saharan Africa, and an estimated 241 million people are infected worldwide as at 2020, and about over 600,000 deaths recorded in year 2021, according to WHO. Of course, drug resistance of P. falciparum has become a worrisome situation for the control and elimination of malaria globally. Sulfadazine, a combination of antiphospholates with sulfadazine, target the parasite's synthesis gene. So this has become, that's why an idea to get drugs that can target this is being developed.
Okay, great. Thank you.
Hello? Hello? Hello?
Hello?
Hello, please. Hello, yes, please. Hello? Hello? Hello?
Unfortunately, I don't think he can hear us.
Hello?
Okay. That is okay. We did get to hear from everyone today, so that was really nice of all of you to be able to present your lightning talks. I can go back to the first page where you can see all the presenters' names. We do have a few more minutes if anyone has any questions for any of the presenters. You can, if you do, you can go ahead and unmute yourself. You can put in the chat. Okay, if not, I just want to thank you all for coming to this lightning talk session, and a special thank you to all the presenters. It was really great to hear about all of your research, and thank you all for joining from all over the world. And I hope that you enjoy the rest of the day and the rest of the talks.
Thank you.
Thank you.
Thank you.
Thank you so much.
Thank you, everyone.
It's a pleasure to be here.
Thanks, everyone. It's a pleasure.